Good evening, ladies and gentlemen. Thank you so much for joining our Eating Disorder Hope webinar for this evening with a special presentation on the Binge Eating Genetics Initiative, a research study about the genetics of binge eating, featuring our special guest, Dr. Tasha Woods-Smith, and Dr. Laura Thornton of the UNC Center of Excellence for Eating Disorders. Before we begin our presentation, I just would like to inform you that you will have the opportunity to submit questions to our presenters at any point of the webinar presentation this evening. We will be having a live Q&A session with our presenters following their presentation. So please feel free to submit any questions through the question pane of your control panel. You can directly enter in your questions there. At any time, we will be receiving those questions and submitting them to our presenters at the Q&A period following their presentation. I would like to turn this over now to Dr. Laura Thornton, who's going to be beginning the presentation for us. She is the Deputy Director of the Anorexia Nervosa Genetics Initiative, known as ANG, and Associate Research Professor in the Department of Psychiatry at UNC. So welcome, Dr. Thornton. We're so glad to have you and Dr. Woods here with us tonight.
Thank you very much. I am very delighted to be here with my colleague, Tasha Smith, to present to you a new study that we hope to roll out very, very soon, the Binge Eating Genetics Initiative, which we are calling BEGIN. This study will be conducted at the University of North Carolina, and this webinar is structured to give you the why and the how of BEGIN. We hope to get you excited about this research project and the science behind it, and then answer any questions you may have. Now, in designing our study, we first reviewed what we know. Binge eating disorder and bulimia nervosa are more common than anorexia nervosa, yet research on these two eating disorders is vastly underfunded. In fact, funding for evaluating the molecular genetic underpinnings of eating disorders has focused solely on anorexia because the perception is that anorexia is more severe. However, we know that binge eating disorder and bulimia are serious illnesses. Our own research has shown that patients with binge eating disorder have more psychiatric and somatic comorbidities than individuals without these disorders. In addition, those with binge eating disorder tend to have increased utilization of psychiatric and non-psychiatric medications compared with matched controls. In research we have not yet published, we found that individuals with binge eating disorder have also greater healthcare utilization both before and after the eating disorder diagnosis was made. Regarding course of illness, we know that diagnostic flux occurs across eating disorders. We specifically looked at the diagnostic flux between first evaluation and 1-year follow-up in 807 patients with a binge eating disorder diagnosis at either the initial contact or at follow-up. So on this slide, what we have here is time 1, which is the initial contact. Time 2 is the follow-up. These bubbles are proportional in size to the number of individuals with each of the eating disorders at time 1, and then the same thing for time 2. The lines that go from one to the other show the transitions that we're seeing from time 1 to time 2. And again, the thickness of the lines proportional to the number of individuals that are going to each of the various other eating disorders. So in this study, we found that 695 individuals, which are those right here, had binge eating disorder at first evaluation. Some of those we had no follow-up, so they didn't show up for a second visit. Of those who did have a follow-up visit, about 40% had a second binge eating disorder diagnosis, so clearly they were still an active illness. 40% didn't received no eating disorder diagnosis. So these individuals were probably in remission. But a full 20% who had a follow-up visit received a diagnosis of anorexia, bulimia, or eating disorder not otherwise specified. Located here, here. And we also observed that diagnostic flux was bidirectional, with patients transitioning to binge eating disorder, right here, from anorexia, bulimia, and EDNAS. Now, this is important for BEGIN because we're evaluating those individuals with binge eating disorder and bulimia because we want to get at strictly binge eating. So knowing that this flux is happening gives us some license to go ahead and combine these disorders in this project. We also know that genetics plays a role in binge eating disorder and bulimia. Family studies have shown that first-degree relatives of individuals with eating disorders are more likely to get an eating disorder diagnosis than the general population. And this is true both within and between disorders. So if you have a relative with anorexia, you're not just more likely to get anorexia, you're also more likely to get binge eating disorder diagnosis. Now, if we look specifically, we find that you are 2.2 times more likely to get a binge eating disorder diagnosis if you have a first-degree relative with that same diagnosis. And this is compared with individuals who do not have any relatives with binge eating disorder. One hypothesis as to why we might see this is that it's due to environmental influences. People in the family have the same access to the same foods, have the same celebrations, witness the same eating behaviors. However, evidence suggests that risk levels this great cannot easily be explained by purely environmental factors. Now, from twin studies, where we compare identical to fraternal twins, we get estimates of heritability. And heritability is the extent to which genetic differences among people contribute to individual differences in binge eating behavior. Binge eating disorder and bulimia are highly heritable. And so is binge eating behavior itself. Although it's not shown here, we stripped away the other criteria and just looked at the large amount of food and loss of control and found, in fact, that 50% of the liability to binge eating can be attributed to genetic factors. The take-home message is that genes contribute to our vulnerability to binge eating. However, because molecular genetic studies to date have focused only on anorexia, we know nothing about which genes or even which areas in our DNA might influence binge eating. Now, what we do know about inherited traits is that the genetic influence exists on a spectrum. On one end, we have traits that are 100% determined by our genes. These things include eye color and medical conditions such as cystic fibrosis or Huntington's disease. On the other extreme, we have things that are 100% environmental, such as picking your favorite sports team. Now, most human traits and risks for medical conditions lie somewhere between these two. Therefore, most human traits, including binge eating, are only partially inherited. The important message here is that having risk genes alone does not determine whether an individual will experience binge eating. The importance of environmental factors should not be underestimated. In fact, the interaction between genes and environment is the basis of many behaviors, and binge eating is no exception. So why are we starting with genetics? Well, we start with genetic factors because, unlike environmental risk factors, the genome is finite and therefore much easier for us to study. In addition, gaining an initial understanding of genes could provide us with invaluable information about how genetic risk may interplay with environmental risk by making certain individuals more vulnerable to certain environmental factors, which can then lead to binge eating behavior. All of this could be the basis for precision medicine for binge eating. Thus, our goal for the BEGIN study is to uncover or identify the biology that makes us vulnerable to binge eating. In doing so, we hope to do three things. First, destigmatize binge eating disorders. Second, encourage research funding and focus. And third, provide information that will lead to more effective treatment options. Broadly speaking, Dr. Cindy Bullock, the principal investigator, designed BEGIN to bring binge eating into the realm of psychiatric genetics. So we will be conducting the largest, most comprehensive genome-wide association study on binge eating disorder and bulimia nervosa ever. And going beyond that, by employing a multi-partner design, we will be collecting a wide array of phenotypic data. Much of the data will be collected using a smartphone app so we can get real-time measures. This means that we have deep phenotyping information using current technology. These are characteristics which continue to make this study unique. Then, by integrating the genetic data and the rich, diverse phenotypic data, we anticipate that we may be able to develop algorithms that can predict risk, course of illness, severity, disordered eating episodes, and treatment response. Now, this has not yet been done. This type of study has not yet been done in any eating disorder. And with all of these elements, we hope to capture data on individuals with binge eating disorder and bulimia that reflects the true complexity of the human behavior. So how do we get to detection and treatment and prevention? By starting with genomics. Well, we review our genome. It consists of approximately 3 billion DNA base pairs made up of adenine, thymine, cytosine, and guanine, which are often referred to simply as here A, T, C, and G, respectively. Within these 3 billion base pairs, there are between 10 and 30 million sites or base pairs where at least 1 in 20 people have a substitution. And what that means is where most people might have an A, the substitution might be a T or a G. Or if a person happens to have a C, someone with a substitution might have a G or an A. When you look at two specific individuals, what you find, so imagine yourself and some random person driving down the highway, is that you have up to 20 million base pairs in total that are different between the two of you at this, when you look at each individual location. So the point I'm trying to make here is that there exists a lot of variation in the genome. And this variation is what we evaluate to determine if certain areas in our genome may contribute to specific traits. So in genome-wide association studies, or GWAS, we zoom in on the chromosomes in your genome and scan millions of sites for the A's, T's, C's, and G's without any prior assumptions. And so these are the different sites, represent the different sites we would be looking at. Now we look at differences in base pairs and cases. Now for our study, cases are those individuals with binge eating versus controls. Those would be individuals with no history of eating, of any eating disorder. We do this one site at a time, and with current technology, we can do this for more than 1 million sites. So if we take a look at the hypothetical DNA segment of 2 individuals, what we find is that you can see that while individual 1 has a letter C here, individual 2 has the letter T. And in our hypothetical example here, 62% of cases will have the less common C, where only 38% of controls will have the less common C. Now, we take this one step further. In this example, individual 1 has an A, and the other person has a G. So in the statistics allows us to evaluate whether there are systematic differences between cases and controls. Areas within the genome that have these systematic differences may be associated with binge eating. In our example, the first comparison potentially indicates an area that might be associated with risk for binge eating because it does appear to be a difference. Of course, we have to measure that statistically. And in the second case, these appear to be very, very close. So probably this area would not confer risk for binge eating. Right, this is all fine and wonderful, but what does the real world look like? Well, among psychiatric disorders, schizophrenia is an example of a GWAS success story and one that we really hope to repeat in eating disorders in the next several years. So when I mention schizophrenia, I want you to be thinking about this as binge eating, because this is where we really do hope to go. Schizophrenia was one of the first psychiatric disorders in which the GWAS method was applied. And what you're looking at here is a Manhattan plot. This is a standard way to visualize the results of the GWAS. The x-axis here depicts all of the chromosomes. And as you can see, the length of the chromosome corresponds to the length that we actually see in chromosomes. Number 1 is the largest, and number 2 is the smallest. And the y-axis depicts the statistical significance thresholds that we're looking at. Each dot in here represents a genomic site in which people have different letters or base pairs. Any dot above the red line, which I'm sorry, this does not look to be red on the screen right now, are considered statistically significant, meaning they increase risk for schizophrenia. In 2009, as you can see, there's nothing above this red line, so no genes increasing risk for schizophrenia were identified. And I want you to note the sample size. We've got 2,600 cases, 3,300 or so controls. Researchers quickly realized that large sample sizes are crucial for GWAS, and this is not even close. So in 2011, they increased the sample size. And now we've got over 9,000 cases and over 12,000 controls. The sample size this large, they found 5 risk genes being identified for schizophrenia. As you can see, we've got 2 on chromosome 6, 1 on chromosome 8, 1 on chromosome 10, 1 on chromosome 18. Then, in 2013, they more than doubled the sample size again, so now we've got about 28,000 cases and controls, and we found 62 genes that may confer risk for schizophrenia. More recently, even than that, we are now up to 35,000 cases, 27,000 controls, and we have 108 schizophrenia-associated loci. All right, this is all great. We found these areas. How does this now translate? If we look more closely at the findings, we see that one of the significant genes is a dopamine receptor. This finding makes sense since current medications used to treat schizophrenia act on dopamine receptors in the brain, thus providing confirmation for the role of dopamine in the biology of schizophrenia. So right off the bat, we can see how GWAS findings relate to treatment and how they can tell us about the biological pathways involved in binge eating. But that's sort of going backwards. We have the drug, now we're looking at the genome. Well, we can also go forwards. There have been significant findings involving glutamate genes. In fact, we've got 3 of them right here. Even though glutamate is also a neurotransmitter in the brain, it is currently not a target for schizophrenia treatment. However, if confirmed by other lines of research, glutamate could potentially be a target for new drug development. Certainly, genetic studies could lead to more effective treatment strategies. Further down the road, we may also be able to tailor treatments specific to an individual's genome, which would be in line with the concept of precision medicine. So this is a glimpse of what we might be able to do with the genetic data alone in this study. Tasha will be presenting the details of what the phenotypic data we will be collecting will be, and will give the basic mechanics of the study. Because the study goes so far beyond traditional GWAS by including deep phenotypic data, the implications of combining genetic and phenotypic data have not been fully elucidated. This is truly cutting-edge science. And I'm going to turn this over to Tasha.
Great, thanks. All right, give us a second to transition here. Can everyone hear me? I guess only Crystal can answer that. All right. Can you see my screen?
Yes, we can see.
Perfect. Okay. So thanks to Laura for that introduction. We'll talk now about the specific logistics of the study, the types of data we're going to be collecting, and how with this next section. So as Laura mentioned, we have a few goals with the BEGIN study. The sort of primary, most important goal is to uncover the biological mechanisms underlying binge eating. We're doing that, of course, with the genomic analyses. The second is to determine the effectiveness of a smartphone app for binge eating disorder and bulimia disorder symptom management. We'll talk about that a little bit more. This is more of a secondary goal, which we'll talk about. And a third goal is to determine whether the addition of a wearable device, in our case, an Apple Watch, increases engagement with the smartphone app and whether that increased engagement results in better outcomes. So as Laura mentioned, the BEGIN study is a case-controlled design. We're looking to recruit 2,500 people with either a lifetime or current diagnosis of BED or bulimia, as well as 2,500 people with no personal or family lifetime history. The no family lifetime history is important because the main goal of the study is to look at genetic risk for binge eating. We'll be collecting data on genes and behavior, as Laura mentioned. The BEGIN study is huge. We're just collecting a ton of data, which uniquely sort of positions our study as a really unusual combination of genotyping with deep phenotyping. So we'll talk about that. So the way that the BEGIN study works is we're doing all of the enrollment, including informed consent, initial questionnaires, screening, those sorts of things, inside a smartphone app. We're partnering with Recovery Record, which I'll talk about in just a minute, for iPhone users. So everything happens inside the app. Once a study participant is enrolled in the study, they're confirmed eligible, they agree to participate. We send them a kit in the mail with a saliva collection for the genomic analyses, a microbiome collection kit from study partner Ubiome, and then all the rest of the study collection takes place inside the Recovery Record app, which for some people in the study will also be paired with an Apple Watch. So inside the app, we're collecting a ton of data. So we're collecting an enrollment questionnaire, which confirms eligibility for the study. We are prompting users to log their mood and food 5 times per day for 30 days, and I'll show you what those prompts and the response options look like. We're assessing current ED symptoms with the EDEQ. We're assessing that at baseline, midpoint, and at follow-up. We are assessing current anxiety, depression, gastrointestinal systems. Symptoms. We're also assessing for ADHD. And for the users who get the Apple Watch, we'll also be assessing their physical activity and their heart rate on a daily basis for 30 days. So why pair mobile mental health with eating disorders? Well, as I'm sure you all know, smartphone use is pretty ubiquitous, especially in the United States. This data, 77% of US adults own a smartphone. I think that's old. Actually, I'll have to check the references, but that's from the Pew Charitable Trust, their mobile research unit. Also, 56% of people with binge-type eating disorders never seek treatment. I actually forgot to reference that, but that is from a study at Harvard that used a nationally representative sample, showing that the majority of people with binge-type eating disorders never seek formal clinical treatment. There are a number of reasons that we could speculate for that. There's certainly so much stigma surrounding binge-type eating disorders. There's lack of insurance coverage. There's also lack of providers with specific training in eating disorder treatment. So we wondered whether smartphone-based apps can help fill the treatment void. If perhaps somebody might seek care via a smartphone app, an evidence-based smartphone app, who maybe wouldn't go see a clinician face-to-face. So we're partnering with Recovery Record in this study, and we're going to be looking at whether, a smartphone that is free, or a smartphone app that's free, that's private, and that has pretty wide reach, whether that can help us improve outcomes for people with binge-type eating disorders. So just a quick introduction to Recovery Record. They're the most widely used and evidence-based smartphone app for eating disorder symptom management. And And also, this is just a research partnership. There is, we don't own any, UNC doesn't own any portion of Recovery Records platform or technology or anything like that. So just to resolve the sort of conflict of interest, if anyone has any, is wondering about that. Recovery Record has an existing user base of over 400,000 people. Recovery Record is used to treat and manage the symptoms of all eating disorders, not just binge type eating disorders. So, in that 400,000 people, which honestly, their platform is probably bigger by now, but there will be a significant number of those who use the app for anorexia. But still, it's pretty large user base. In 2015, they polled about 100,000 of their users with binge type eating disorders and found that a full 50% of them had never sought clinical treatment for their eating disorder. Interestingly enough, a full 33% of those who were polled had never even told another single human being that they struggle with an eating disorder. So clearly, people, despite being actively ill as well, so Recovery Record had administered an EDEQ to those people and found that even people with very severe symptoms had either never told anybody that they struggled and had never sought treatment. So clearly there's a need that apps like Recovery Record are filling. And again, it's free. And the thing about Recovery Record is for those users who do seek clinical care, Recovery Record allows users to connect with clinicians, whether that clinician is a therapist or a dietitian or both. So Recovery Record, in partnership with UNC for the Begin Project, they've built a 4-week program that's CBT informed. It's built for self-monitoring. They're integrating it with the Apple Watch, and it prompts the user 5 times per day to log meals and mood. So that's 3 main meals and 2 snacks every day, and they give weekly personalized progress reviews. I'll show you a little bit about what it looks like. So one cool thing about Recovery Record is they give you the option to receive sort of discrete prompts, and this is what the prompt looks like. This is Recovery Record telling you it's time to log your breakfast. If you respond to that push notification, it takes you to this screen, which it's very much, and it'll look a little bit different from this. This is the last iteration that we got from them, but you'll see that it's less about like, tell me what you ate and what the portion size was and whatever. It's really prompting the user to check in with themselves and just notice how they're feeling, what their emotions are like, and then it'll prompt food-related things like, which meal was this? Where did you eat it? Did you eat it with company? And this is just scrolling down that same thing. How long ago did you eat, with the goal of trying to get people to log as closely as possible to actual consumption. And then way down, it tells you, okay, what did you eat and drink? But again, this is open-ended. We're not asking about portion sizes or anything like that. And then the user rates it as inadequate, adequate, or excessive. And then it asks you, okay, how mindful was this meal? Did you restrict? How strong is your urge to restrict? Did you binge? How strong is your urge to binge? And then there's an open-ended section just for thoughts. So with Begin, we'll be using Recovery Record and genetics and microbiome and just a battery of psychological tests to really bring genotyping with very deep level phenotyping. And we're hoping that that this, measuring body and behavior at the same time, that this will really help level the eating disorder research field, as Laura mentioned, bringing binge eating disorder and bulimia to the forefront. We're hoping for, this is really a hypothesis generating study. We're collecting a ton of data, and then we'll use the most advanced statistical techniques to look for patterns in the data. BED and bulimia are just so understudied. We're so eager, and we're super grateful to be just on the edge of launching this. Laura and I haven't mentioned, but the big study will be 2,500 cases and 2,500 controls, but we're starting first with a very small pilot just to make sure that everything works. The larger study, the 2,500 cases, 2,500 controls, probably won't launch until this fall or maybe late summer if we get lucky. So just a few references and a thank you to our partners, including UBiome. And that's it. If you want to get involved, I included a couple of links which you may not be able to click on, but I'm sure your organizers wouldn't mind emailing those out. And if you have additional questions beyond what we're able to discuss here, you're welcome to email me at that email address there.
Great, thank you so much, Dr. Smith. And just a reminder to all of our attendees who are with us now, if you have any questions that you'd like to ask to either Dr. Smith or Dr. Thornton, feel free to submit those now through the question pane of your control panel as we transition into this Q&A period. And we did get a couple questions, if that's okay, if I can go ahead and read those out loud.
Yeah, absolutely.
One of our attendees is wondering if the study is parallel at all to the Angie study, and if you have any updates with that study as well.
Yeah, I can field this question. Certainly, this study we began because of the Angie study. So initially, it started out as a general study of genetics, but we've really expanded it far beyond what we're doing with Angie in terms of the phenotyping. So yes, we're doing the genetic part, but then we've gone even further, as Tasha was explaining. In terms of where we are with the Angie study, at this point, we have finished our data collection, and we are processing all of the blood samples to do actually the GWAS studies. We hope to have the initial results within the next 6 months, but then, of course, data cleaning and analysis has to happen. I'm really hopeful that by possibly as early as late this year, we'll have something to present to folks. I'm not making any promises on that, but that's the hope at this time. I will say this, we did meet our goals. We have over 13,000 cases and 13,000 controls, so individuals who submitted blood samples for the study. So that's pretty impressive for eating disorder.
Thank you so much. Another question is wondering, over what period of time will the BEGIN study take place, and how many participants are you hoping to study?
Yeah, so our initial pilot, which is about to begin, will study only 500 participants just to make sure that all of we're collecting a lot of data, and we need to make sure that those data streams are managed and we can integrate them effectively. And then once that is done, we'll move on to the full study is 5,000 people. So 2,500 people with a lifetime history, a current or a lifetime history of BD or BN, and 2,500 people with no family or lifetime history of an eating disorder. So 5,000 total.
And we hope to manage to collect this in less than 2 years. We anticipate that at least for the DNA part, it will be much easier than it was for Angie, because we're collecting spit instead of blood. Yeah. And then all the rest of this stuff, we don't have to do a personal consent where we have to talk to them. So we really tried to streamline this to make it very easy for participants to enroll, to participate, to do what they need to do, and for us to get the data that we need. So we're hoping that because we've done all of that, that this will be a much faster data collection.
Yeah, and I don't know that I made this clear, but we're recruiting from the existing user base at Recovery Record almost exclusively. Although there will be some additional avenues of recruitment. Also, I see somebody's note that they could see my notes page. I'm sorry. I was working off a dual monitor, and I think it shared the wrong one. Sorry about that.
Oh no, you're totally fine. Thank you. And thank you for addressing that question. Um, we also have attendees wondering if participants who may actively be in treatment for BED, are they able to participate?
Yes, they are. Yep. Even if they're actively seeking care now, even if they're connected with a clinician inside Recovery Record, they can participate.
Great. And just another question kind of in follow-up to that, are there any criteria that might exclude someone with binge eating disorder from participating in the study?
Yes, there are some, and it's actually kind of a long list. There are a number of things that we record but don't exclude on the basis of. There are some exclusionary criteria, but I don't have them right in front of me. But those tend to be focused on the specific research questions that we want to ask. Obviously, we try to minimize exclusion criteria just so we get the widest possible participation in the most representative sample. But because we're measuring things like the gut microbiome and such, we do exclude on the basis of some GI conditions. We exclude if the participant rate's pretty high on recent suicidality, things like that. But on the whole, we try to minimize exclusion criteria.
Yeah, and of course the participants have to be 18 years of age.
Oh yeah.
We're not taking children. We're not getting parental consent for a child. And then things like if you're on current antibiotics, because we are studying the microbiome, we then give you sort of a wait period before you can enroll. So you're off the antibiotics for a particular period of time. And like Tasha said, certain GI problems, if you've had any type of gastric surgery, that's generally an exclusion, just because of how it's interacting with the microbiome.
Great. Thank you for sharing that information. Another question from one of our attendees wondering if the smartphone can pick up changes in heart rate and adrenal responses.
So the smartphone can. This gets a little bit complicated. So for the users who get the Apple Watch, and not everyone right now in the study will receive the Apple Watch, that may change in the future, but for those who get the Apple Watch, we can sample heart rate at least once per day. If we can get multiple samples of the heart rate per day, then we can definitely look at heart rate variability, which is important. We're hoping to do that. Things like adrenal responses, no, we don't have a way to measure that presently. But heart rate we're hoping to get from the Apple Watch.
Thank you so much. Another question, how will you separate the results with the increased multiple diagnosis like depression, anxiety, etc., versus the BED or BN when analyzing the genome? Does that make sense?
Yeah, and Laura's more the geneticist, but I can take a stab at that. So for things like anxiety and depression and ADHD and the other psychological things that we're measuring, those tend to be covariates in the analysis. So they help us to control for those conditions. They can help us stratify by extreme scores. Yeah, but when we actually look at the genomic analyses, the whole purpose of having a case control design is so that we can look at people with a current or a lifetime history of a binge type eating disorder and compare them to people with with no family, personal or family history of an eating disorder. And we can make that comparison. And usually things like anxiety and depression and whatnot are just covariates in that analysis.
Although the concern is a real one, so the person who put this forward, because we do know that depression and anxiety are increased in binge eating and compared to the general population. So it is something that we are aware of, and it is something that we can control statistically. And certainly, once we get a large enough sample, we can start separating that kind of stuff out. At this point, though, we're just looking at binge eating and not, and we have all of that other information, so we can then use it later on.
Great, thank you. And just the last question.
Yep.
Okay, sorry. Last question that we have is, what do you hope to see in terms of advancement with binge eating disorder treatment as a result of this study?
That's a great question. And I think some of the results from the genetic analysis, so when you get into the pharmacology, are going to be so far down the line that that's not something we're going to see immediately. Okay, so I think the things that we're hoping to see is the feasibility, the acceptability of using the app on the smartphone to see if that's truly helping. We're going to be, because we're monitoring these individuals over 4 weeks, we're going to be able to see if the engagement actually truly does reduce binge eating episodes and other behaviors that go along with these eating disorders. But unfortunately, I am not a clinician.
Yeah.
The best to answer that.
Yeah, no, it's Begin is really unique in that regard, because the genomic work can identify biological pathways that for which treatment options may not emerge until the very long term, as Laura said. But the behavioral aspect, using a smartphone app that is evidence-based, built for symptom management, pairing that with a new emerging technology like the Apple Watch, those are the types of things that might help us, evaluate and improve those treatment options in the near term. So we have a sort of short-term and a long-term goal to evaluate and improve and look for new treatment options for BD and BN.
And I think one of the things that was not made clear necessarily in this webinar that people just don't realize is that we're going to be getting so much data from the gut microbiome and have the genetic stuff and all this phenotypic stuff that we actually are going to have to apply machine learning, and there's probably statistical methods we're going to have to use that haven't even been invented yet that can possibly give us algorithms, as I said, to predictive algorithms to look at course, look at severity, to hopefully help with treatments, all of that kind of stuff. But that's the exciting part about this, is that we are really at the cutting edge of this, and we don't know how it's all going to come together.
Yeah, as Laura mentioned, so just to unpack that machine learning element, we don't advertise that too much, because we're still not sure what these data are going to look like once they roll in, and whether they'll be sufficient. But if users log their meals and their moods and their binge episodes, we can record that in real time and split that data set into just this nice, robust data set that we can maybe learn how to predict when binge eating episodes might be, forthcoming. And in the future, we might be able to deliver some real-time intervention via a smartphone or an Apple Watch or something like that, that can help prevent and divert those forthcoming binge episodes during high-risk periods, potentially. Which is, I mean, that's another like really exciting thing. But I would consider that a more like medium-term option, that we're hoping to get at with Begin.
Thank you so much for sharing. We actually had one last question come
Yeah,
in.
sure.
Is it possible for a person who's also in treatment to have their clinician collaborate via the app? So, you know, if it's a therapist or dietitian who's working with this person who's also in the study, that, you know, they're all kind of involved together.
Yeah. So right now we don't collect any data from the clinicians. So if a user is connected to a clinician, be that a therapist or a dietitian, via recovery record, we don't then go and pursue that clinician for additional data collection. We could perhaps if we got some additional funding and whatnot, but so in terms of a clinician actually participating in the study, they can certainly encourage their client or their patient in the study, and of course supplement the treatment that they're getting via the app. But we don't currently have a way for clinicians to participate in the study, but that's a great suggestion.
Great. Well, thank you so much, ladies. This was a very interesting and fantastic presentation, and we just appreciate you both taking the time to share this exciting research, and we're excited to see what comes out of your study.
Well, thank you.
And I think you already have the website up, Dr. Smith, but for anyone who's wanting to get updates on the study, is it just at the unceatingdisorders.org?
It is, yeah. When the study goes live, we will have a study webpage, but for now, it's just unceatingdisorders.com or .org. If you click Join the Cause, it'll take you where you can join the SEED Research Volunteer Registry, and we'll send regular updates there once we get started.
Wonderful. Thank you so much for all this great information. We appreciate you both taking the time to be with us this evening. And to all our attendees who were able to join us live tonight, thank you so much for your participation in this webinar. We invite you to visit our website, eatingdisorderhope.com, where you can find out our webinar schedule and see the future webinars that we have upcoming that you're welcome to register for as well. So on behalf of Eating Disorder Hope and our presenters this evening, thank you so much for joining us, and we hope to see you soon. Thank you again, doctors. Take care.
Thanks.
Bye-bye.
Good night.